Even though I have a lab starting on Monday after your class here, the 11 to 12 folks, I said that we could, if they needed a few extra minutes, I don't mind going over and opening the anatomy lab. I would do the same for you. I can just tell my advanced physiology lab we'll start 3:15 or 3:20. It's not. It's probably more of a lecture lab than a hands-on lab for them anyway. And they've had to put up with me for a year already, so they know me. So if you're 65 questions, you can have the full hour if you want. I don't know if anybody comes here after it, but if they do, we can scoot over next door. If nobody does come in here, and I'm not sure if somebody does. I don't know if Dr. Maurer comes in here after. Maybe I'll ask him if he does. But if not, we can scoot over next door to the A&P lab because hey, I never leave home without it. Moving on. So that's the broadcast news event of the day. We all heard that out there. You don't talk back the virtual, 10 days. I'm happy with that. So I'm going to pick up where we left off, and really today we're going to look at in a little bit more detail, even though we went over the numbers for STEAM, but physical methods of control. And I think that's really where we start with here. I want to make sure I have both PowerPoints. I have Callan's PowerPoint and I have the Nestor PowerPoint. And I'm gonna maybe flip-flop a little between them so you get information on both, because there's some better illustrations in Nestor about really the autoclave. And this is very apropos, whatever you're going to be doing. For most of you in some type of clinical environment, I think you do need to be, why am I getting back to the beginning? I don't like that. Here we go. Okay. So heat as a method of control. Why do we use the first sentence tells you everything you need to know. Remember that thermal death curve. True. When you heat things, the lethal temperature is so close to the optimal temperature, it doesn't take much more to kick it off. And effectively what it does is you're going to see it denatures the proteins. That's the goal. Freezing or refrigerating things can be desirable for certain things you want to preserve, realizing that the proteins may start to denature, but it's not true. Denaturing is a permanent change. So they're just going to become less active, or relatively speaking, inactive. But once you bring them back to temperature, hey, we're back ready to go. So when we talk about it, there are some things that you certainly could sterilize by boiling, but in general, not acceptable. The beauty of moist heat, just to reiterate the point, is it's superheated steam. Tiny water molecules that are super hot and super fast and very destructive. And it doesn't matter when you have things stacked on top of each other, there's always space for those molecules. So you don't have to be as cognizant of how you arrange them. You basically just throw them in, a big stack of them, and they're sterilized. That's what they do in the operating room and in the surgery, what they call central surgical sterile supply. So they do those kinds of things. So it's very advantageous to do it that way. Where dry heat doesn't penetrate. That's just absolute temperature. And because it's not water vapors, it will still cause proteins to denature, but not in areas in contact, because the heat effectively won't get through, if you want to look at that way. So there's a lot of useful temperatures. We'll go over that. In the lab, you're incinerating is what you're doing. Incineration is very reliable. That hazardous waste in those red bags, it's all incinerated wherever they are permitted to do so. They used to not be able to do it in Pennsylvania, where you had to be carted to Ohio. And that's, again, part of the record-keeping thing that's so important. But animal carcasses, a vet that's had an animal they had to put to sleep because it was sick or very old. A lot of veterinarians will have their own system. They incinerate it, and obviously flaming your loop. So moist heat, the advantage, the temperatures certainly not as hot as a flame or an oven, and you don't have to generally, with the exception of a loop, which is relatively quick and not a large-scale operation, it's very, very effective. and it's microbicidal. And dry heat, same thing, dehydrates the cell, removes the water, denatures the protein, alters the, yeah, and there are some proteins that have a certain amount of stability. But when they get to a certain temperature, it's all over. I mean, we consume proteins, we heat them up and denature them to some extent, and we basically kill the bacteria. We cook them. Okay. I mean, there's different, and that's why you see that information on the package for certain, internal temperature of 160 degrees, because basically you could be pretty darn sure that none of those nasty bacteria that are going to give you food-related infections or viruses are going to be there. Right. So, I mean, that's my single most important piece, almost. My microplane grater is, I think, my most important piece of kitchen equipment. The other one is that little digital thermometer. Oh, hello. Instant read. And that's what I mean. When you denature something, it's done. The shape can't change back to resemble this either partially or totally. It's just done. And these are the numbers. And so the main number is right up top, that 121 degrees Celsius for 15 minutes. Authors will tell you 20. I don't know which one's on your exam. It's the next set of exams. It's either 15 or 20, but it's not both. And if it was both, I'd have to take both answers, and I certainly would. And all that means is that when you pressurize steam, meaning you have a certain amount of water and you're heating it up in a closed system where the volume can't change no matter how much steam is produced, all it can do is superheat. Okay, and that's really the thing. I gave this analogy. This is a chef trick. Yeah, I don't know, because here they tell me in dorms all you can have is microwaves, right? You can't have an Instant Pot or a little range top, like a little burner top, right? You're not supposed to. I say nothing. I know nothing. You probably have one. Don't ask me no questions. I won't tell you no lies. Okay. See, I'd have a whole apparatus. I'd have my propane powered flat top. We're doing burgers. We're doing breakfast. We're doing everything. That was our mutual Christmas present to each other. That's us. 101. And so if you put water in, like you're going to make a cup of tea or a cup of soup, something, and you put it in there for 3 minutes in a cup, and maybe you were in a hurry, you yanked it out fast, you probably got splattered. What happens is the water has surface tension. And what's happening to that water in the microwave, the top of the water, the molecules are holding on to each other. And when you move it, you break the surface tension, superheated vapors, steam, come up, and you can get a burn. It's not a big burn, but it's a burn. So what chefs do is they put a glass rod in, like that big Pyrex, cup, 2 cup, 4 cup things in their microwave to heat water, because it doesn't allow the surface tension to form. So when they're in a hurry to pull it out, they don't have to worry about it going all over the place. So that's the idea. That's what superheating is. All temperatures, moving those molecules around fast. The 134 degrees for 3 minutes is the flash sterilization number. Drop an instrument out of the sterile field. It's relatively clean. It may need to be used. They have a sterile corridor that runs behind all the operating rooms. They're going to have a device somewhere that is set to 134. They'll put it in for 3 minutes, bring it out, dump it in some cool sterile water or sterile saline, and you can use it again. So there are ways that you can take something that's already relatively sterile, and despite the fact, because the minute, again, it leaves the field, not longer sterile. It's a very limited window. Dry heat, there's a lot of numbers. The consistent number that most people will be between 160 to 200 degrees Celsius. That's 350 to 400 in your basic oven, more or less 400, 4 1/4. And again, because it doesn't penetrate, you have to get a sheet pan, cookie sheet, and just single file, no overlaps, no touching, and about an hour and a half, they'll be sterile. The difficulty is they're very easy to recontaminate. How do you get them out of there? Okay, you have to have sterile gloves on once they cool. Once they start to cool, if you take them out, they might get bacteria. what you'll see in an operating room or in a hospital or a medical facility, they have like a paper and plastic. The plastic is designed to withstand that high heat. Paper portion is permeable to steam vapors, but microorganisms can't get through. Remember, water's a tiny molecule. And so they'll seal them. They'll put a retractor. They'll put a self-retaining retractor, an instrument, because you could, through the plastic, you know exactly what it is. And you can sterilize it. Once it cools down, you can just lift out the pouch with your fingers. The inside is sterile. And it'll stay sterile for X amount of time, unless you open it. And if you use it, you have to re-sterilize it. There's a whole lot of it. We did a lot of that stuff. 90% of the procedures I did were office-based. Only about 10% were hospital-based. So we did all those things. I had a sterilizer, my wife, BSN RN at the time, active at the time. My first wife, she trained everybody how to do this, how to wrap it, da-da. So I had a certain, advantage in that regard. Let's see if I can get where. So again, endospores are the judging part. Okay, so its temperature is ordinarily above boiling. Vegetative cells, the rest of them, that's when you start getting into the variation of death times, and that's what Pasteur studied. And again, he came up, as you might recall, and you should, roughly 62 to 63 degrees Celsius. It's around 140 degrees Fahrenheit for 30 minutes. That eliminates most vegetative bacteria and viruses, and therefore eliminates certainly the most likely organisms for consumable foodborne illness. That was the whole thing with pasteurization. And you'll see some other stuff that's going on. As you can see, and even if it was an endospore form in a vegetative state, not the endospore state, you clobber it that way. It doesn't matter whether they're pathogenic or normal, non-disease causing, similar for other microbes. A little bit more maybe for viruses. That's why we get 10. You're not so worried about those for foodborne illness for the most part. The ones that could be there, most of those, how shall I say it? Most of those viruses that are gastroenteric in nature are not in this category anyway. And this is the way they were calculated. This is what I was telling you about before, calculating the time and what the point is where you can get rid of the microbes. And the Nestor PowerPoint has a chart that shows you how they configured it. It's not a big deal. It just shows you the exponential nature of how they died that I commented on before. So the whole idea of perishable substances, whether it's dairy, whether it was Pasteur's wine, whether it's something you're going to eat, is to maintain what makes it appetizing. Its flavor, its texture, etc. That's there. So by definition, pasteurization or boiling water or any of these more exotic pasteurization methods are effectively disinfection. Okay. And if you live in an area, maybe you've had it around where you live where they had a problem with the water supply or something broke, and they said there's a boil water warning for the next 3 days until we get things fixed. And then boil whatever water you were going to use to cook or to drink, boil it for 10 minutes and let it sit and cool down. Then, assuming you didn't have bottled water or something like that. Because that's what you do. It's and that's what's the first thing to go when we have, the next snowmageddon coming that lands an inch. We had this year. Your toilet paper and water disappeared. You're going out with hoarders. Okay. So boiling water for 30 minutes, surely, but where's the water going to be? It's gonzo. Maybe you can have like a kettle and adjust accordingly. Okay. So this is typically pasteurization. There are some consumables that are not liquid, but it's not essential for us to go into what they are. But it's comparatively easy. They're for spoilage and illness, and that's what pasteurization is. Retaining liquid's flavor and nutritional value. And then we'll get to the autoclaving note in a second. The whole idea from why we utilize other methods today, and I wrote on the board for you that HTST, high temperature short time method, where you crank up another 10 degrees, roughly 18 degrees Fahrenheit, and only have to do it for 15 seconds, is just that, time. High temperature short time. You get whatever dairy brand you use, whether it's milk, whether it's 1%, 2%, half and half, Land O'Lakes, whatever, that is all pasteurized by that method. If it has a higher fat content, ice cream, another 10 degrees. So now we're up to roughly about 176 degrees Fahrenheit for about 20 seconds. Again, fats inherently are a little harder to penetrate through, need a little extra temperature boost. But for that limited amount of time, you're not going to break stuff down. That's the whole idea of the short time. And again, in industry or commercially or for food, it's all about dollars. So you're trying to do something that's going to be reliable without causing, how can I be efficient and do it with still maintaining safety levels? Because heaven forbid, lawsuits kill companies when there's contaminated stuff. They're in big trouble. And then we get into the autoclaving. And again, we use everything that you basically use in surgery, with certain exceptions that are heat-sensitive materials. When we get to gaseous usage or poison gas called ethylene oxide, for instance, like gowns and gloves, needles, and things, most of those little things that those burst open packs, those are gas claved. So we're using poison gas, the gas dissipates. The instruments themselves, most of the hospitals will, like we did at our office, will stack the gauze pads, wrap them appropriately, sterilize them. Rather, because if you're buying an individual sterile, like you might go at the grocery store or at the pharmacy, way too expensive than you buying package, big units of them with cases with thousands of those. And you just put as many as you think you typically need. Sterilize a bunch of them. If you need more, hey, I need a few more sponges. I need more Kling. I need more Weber, whatever you're using for that. You'll see here in like the Petri dishes you're using, those plastic ones, are gas sterilized. When we pour the plates in the micro lab before your lab, pour them like one afternoon. We open the plates. The outside is non-sterile, but the inside, we open it just a little bit, pour sterilized liquid and close it. Boom, done. The plastic is designed to withstand that hot liquid. It can't withstand the autoclave. There are special plastics that do. So that's part of it. So this is really the nugget, and I'm going to flip over to the other here. After I do the dry heat, Bunsen burner, you can see how hot it is, 2,000 degrees Celsius. Okay. Again, a variety of incinerators higher. And there's ways you can do that. If you see like the classic pizza ovens that are brick with wood in the middle and the dome, you can concentrate back the heat. That's why they're, the really good ones, 600, 700. It's pizza. Who doesn't like pizza? I mean, for years, people have been bringing them over brick by brick from Italy to be able to do, that's the way they do, in the high-end restaurants like that. So hot air is different. As I said before, I lean more to that 2 hours, 180 to 200. I think would be better numbers for that. I'm going to show you some pictures. I am going rapidly because I want to give you some time to go over some of the questions, and I have a few more minutes to do so. And here we are again. This is the same area of PowerPoint that we were looking at before. This is the Nestor PowerPoint. So again, doesn't sterilize because the endospores. High temperature, short time. We'll get into ultra pasteurization. You might remember I said juice boxes, drink boxes, little things you peel open for creamers, flavored creamers and stuff that most people are very familiar with. What they do is a specialized container. They sterilize the container with vapor phase hydrogen peroxide because it's superheated, super concentrated. Sterilize the container. Then they superheat the liquid for a fraction of a second. Pour it in, seal it, boom, done. If you are unaware that most of the time, if you're in Europe, maybe notwithstanding England, but if you're in the continental Europe itself, and they buy food every day. There's no Mega Mart with the giant refrigerator system. They don't trust refrigeration. They buy things every day for the next day or two. They don't have a big thing like that. There's no weekly shopping list. The refrigerators are slightly bigger than your dorm refrigerator because the only time you put things in that are perishable is once they're open. They'll buy milk and dairy in packages, which you can get here. Once they're open, you refrigerate. It's a whole. I mean, they don't trust refrigeration. Just we make stuff fresh every day. That's the way we're culturally different than where we are here in that regard. So it's you go down to they all have open air markets. If you're going to make a wonderful mushroom dish and the mushrooms look a little okay, the eggplant looks good. We'll just make an eggplant dish. It's not like I have to make this recipe today. It just doesn't work that way. This is a better illustration of an autoclave. Okay, and basically it's a sealed chamber that's there. You have a certain amount of water that's in there. You replace it, clean it out after you use it. It has basically a rack that separates from the water. The heating element is here, and it creates steam. And you could have liquids upright. You could put aluminum foil, works fine. You know, or you could put it this way if you're doing glassware. By and large, we put boats, stainless steel, perforated on the bottom, and throw clean instruments in and sterilize them. And you'll have, okay, this is your large bone tray. This is your chest tray. Nurses sit there and make cards about what you use. And now, okay, this is Dr. So-and-so's preferred bone tray. Or this is their joint replacement tray. Whatever you're doing. So all those instruments are in a giant pack. They open it, ready to go. That's how that system works. So the water is preset as to how much it is. And basically, as you increase the temperature, you know when it reaches the critical temperature because you're interested in pressure. Again, the ideal gas law, and this is just like closing a door on a naval ship or a submarine, which we know you can. How many places you have no idea how many places on a ship say, do not open this door until you close and dog down the other door. It's awful. I'm walking with my kid when he's a junior officer. I walk over a couple of big ropes on the deck of the cruiser. He goes, don't you do that. And as I'm walking through one of these openings and I have to bend through, he goes, dog it. I knew how to respond to him. Yes, sir. Awful. Now, because anything tragic would happen, that isolates the compartment in such a way that the other compartments, whether it's radiation tight, watertight, whatever it's designed to be, which I know very little. This is what it looks like. So if you go back, and I don't know if they had the picture. Normally these go in, again, on that perforated tray. This is autoclave tape. Autoclave tape, once it's exposed to heat for the appropriate amount of time, has an indicator in it, and you see these stripes. Maybe you've seen them in the lab. That does not mean it's sterilized. You could assume that, but it doesn't mean it. What it means is it was exposed to heat sufficiently to alter the chemicals so they turn black. Sterilization is biological, not chemical. We did this. These are called Dennison wraps. Water vapors get through them. We double wrap them. So we open up that bone pack and put it in there. And if the doctor says, I don't need those, they just won't open it and it's sterile. So you don't have to keep re-autoclaving. If you haven't opened it, it stays sterile. It's the Band-Aid. Contents guaranteed sterile unless opened. And they have a certain shelf life. They can do that. Inside, with a mountain of like this, of stacked instruments, we'd put another indicator tape that had an O on it. If the heat got there, a K. So that was another chemical indicator inside the pack. So you knew that the heat penetrated. Again, not guaranteed. Here is the guarantee. Okay. Every hospital, when they run a big batch, every time they run the autoclave, they put those indicator tubes in. They contain that Bacillus stereothermophilus, which is not a pathogen, but it's the hardest one to get. And then they take it out. There's a way of exposing it to broth inside the endospores and incubate them. If they change colors, typically from this yellowish to pink, it means that the endospores are growing. That's bad. That means the contents weren't sterile. So most hospitals will do those packs and not release them for 24 to 48 hours. They'll shelve them and they'll wait until the indicator means that the unit was working. It's just a way of calibrating. If it isn't working properly, they don't have to get fixed and reorder claims. And documentation-wise, you have to have all those checked off. Read the indicator die, this date, this hour, batch A, batch 41 can go out. The documentation is an important part, particularly so in a hospital. Commercial canning is about botulism. It's a little bit different, not a big deal. Dry heat, again, we talked about. Okay, an hour and a half, roughly 400, works great. They use it for a lot. Incineration you use. We're almost done. Filtration is used when we know specifically like yeasts are in it. So certain like alcoholic beverages, they would cold filter them rather than heat them. I mean, Miller and Anheuser-Busch were doing that. Yeah, maybe it's a marketing tool, but depending on people who brew, might do that. The filter you work with the most, that high efficiency particulate air, nearly all, not tiny viruses. If you're in a room where you have a patient with tuberculosis, they're going to have an exhaust fan pulling that stuff into a filter and pulling it out of the room. It's almost like reverse isolation. You don't want that to, as you walk in, to somehow get to you and you bring it elsewhere in the hospital. You do that when there's a lot of risk. I always tell the same anecdote. My kid, when he was a year and a half, was at the old Children's before they built the one over in Lawrenceville, and was having a GI workup because he had allergic problems with his stomach, it turned out. And he was staying at that time. They would stay. Normally it would be a same day type procedures. We found out the play group he was in at the local Y, there was an outbreak of chickenpox before immunizations and things like that. Immediately you tell the doctors and the nurses. All of a sudden they put him in that reverse isolation. Whenever he went down for procedures, he was this year and a half old baby, now almost 40, covered with, mask, gown, cap, little baby gloves. Just to take him back. When I had to go to the bathroom, I had to wear that and then take it off and change it before I got back in the room. I basically spent 12 hours in the room because I did the overnights. And remind me, we'll start with radiation. I don't want to short shift you. There's not a lot to say about it, but I'll start with radiation on Wednesday. Because now I'm at that 20 minutes or so left. Does anybody have any test-related questions? Or you want me to just do another little go-over for fun? You're cool. Anybody have questions? Like I said, we had about 6 students had questions from the study guide, and then I did this little review, but I'll do it here. So I had separated them, so there was a separate one for their questions. And this one I'll just keep running because I didn't want to show you the questions. That's why the good Lord made cell phones. Okay. Let's see it, baby. I got it. All right. The first 10, 12 questions, say 10 or so, are all about chapter 1. Chapter 1 is maybe 10, 12, 15 questions tops. So it starts with historical figures. Semmelweis and Lister, remember Semmelweis, puerperal sepsis, Lister, antiseptics. Okay, all that in surgery in England around contemporaneous with Pasteur. Pasteur, germ theory is what he felt was causing the problems. We do today. Things we can't see in the air. He was combating the whole idea of basically talking about spontaneous generation. Like there's some vital force in the air that could make an inanimate object, this can, come to life. Yeah, we look at that, we laugh about that, but that was what was the perception. Evil, demonic, whatever word you want to use associated with it. So he was about, and then all those experiments with the flasks and things like that, they were all about that. Pasteur was about therapeutics. He was clinical, vaccines, first vaccine, even before he knew rabies was a virus. Treatments, just like trying to treat that one, all of that. Where Coke was all what you're doing in the lab, letting out things, cultures, growing things in pure culture, all that stuff. That was Coke was much more all the organisms that you're using today in the lab. He grew initially 120 years ago and refined those methods and those Koch's postulates you'll see again, like the chain of evidence for disease, which we don't use today. We talked about genetic engineering. One of the questions was on plasmids. Plasmid is a small circle of useful, because it has resistance, or it can move things from point A to point B, genetic features, but is non-essential, meaning if it goes away, and they do, the bacteria doesn't die. We splice information in DNA on that plasmid, genetically modified information. We make our DNA a little strip, splice it to the plasmid, and use either very receptive E. coli or yeast cells to basically make human insulin, to make hepatitis B immunizations, that type of stuff, human growth hormone. That's what they do. Those are like prototypical things for that are there. I talked about the emergent and resurgent. Emergent diseases, mostly mutation. The other nugget is we're in areas where we didn't used to live. And it's not that the diseases are new, but our interaction with them is new. Living in the rainforest, people like that. If you look around here, the same thing. We built into a lot of areas that were wooded, and that really, even though Lyme disease was around, didn't have a name for it, we increased it. Where resurgent is a disease that we thought was either eradicated by vaccines or by other methods, and now it is making a comeback. And again, measles, because of vaccine laxity. And the other factors are aging, population's getting older. You've got one here. When I was a kid, men lived to, when I was born, I think the life expectancy for a man was 69. I know it's hard to believe, but I have passed that. And again, none of you look at all. You're just laughing about it. I didn't like her the last class we had. Okay, so it changed. So life aging, and there's a whole subset of different things that began to redevelop. Measles, vaccine laxity, traveling all over the place. You tend to, even you go to an area that wasn't eradicated, and you picked it back up, and immunosuppressant drugs and immunosuppressant diseases like HIV/AIDS. And that's where tuberculosis kind of reared its ugly head when we had it licked without a vaccine, just by identification and treatment in a very reliable fashion. And again, what makes bacteria unique? Peptidoglycan. Unique means it's nowhere else. What makes algae unique? They're the only protozoan agent that has chlorophyll. Okay? There's no other eukaryotic microbes that have chlorophyll. Okay, plants have it, but they're not microbes. Fungi and yeast don't have it, although they have a cell wall, like algae does. Protozoans don't have it because even though there can be water-based like algae or land-based like fungi, but by definition, they may have pigmented substances, but not chlorophyll. So things like that are unique or distinguishing factors, really important. Bacteria and archaea. We had a question about the domain theory. Domains are developed. The theory supplanted animal and plant kingdom when we started to have molecular biology. You're looking ribosomal RNA, the 70S and 80S ribosomes, and DNA sequencing. And the authors came out with two prokaryotic domains, archaea and bacteria. Bacteria, they're very similar. Single cell wall, but different because the cell wall has peptidoglycan and bacteria, making it unique. Not archaea. Their size, shape is roughly the same. Where they live. Bacteria more or less everywhere, but archaea only in extreme environments. That's noted. Non-cellular, non-living, or acellular forms like viruses and prions. Viruses, RNA or DNA, never both, because living things have both. And they have a protein enclosure at a minimum. Prions, also similar in a lot of ways, but no genetic material. They're just misfolded proteins. They have distinct chronic diseases all over the place, effective for species. All of that type of stuff may be true, but at their heart, one is just a misfolded protein, the other has some type of genetic material and protein. So again, there's a certain art to simplicity. Viruses have the ability to infect all living things. You and I can get a cold. A bacteria can be altered by a virus. If it's living, bacteria, archaea, or eukarya, all of those, same thing. Okay? Living things have a semi-permeable membrane, the old phospholipid bilayer. The uniqueness is semi-permeability. Some stuff can get in, some stuff can't. It's like a gatekeeper. Do not confuse that, because that's the cell membrane of any bacteria, the outer membrane. We call that one the inner membrane. In gram-negatives is different. Phospholipid on the interior leaf, but that unique, again, LPS or lipopolysaccharide layer outside. And why is that different? Because we don't see it anywhere else. So the lipid component, if it gets where it shouldn't in us, is immediately responded to. We call that toxic. Our immune system views that as an invader. If it's sufficient numbers and a sufficient type, it can be deadly. It has that strand, that O antigen or O polysaccharide projecting from that lipid A. That doesn't really add to its nastiness, but it's how we can use that information to identify it. So it's a unique identifier. It's like its driver's license. We want to look at it that way. So that way we know whether it's just good old-fashioned E. coli used in the lab or O157H7, the H has to do with flagella antigens, that causes life-threatening diarrhea, kidney shutdown. Okay, so more about that down the road. That's the only advantage to somebody who's not as young, because I have a lot of life experience that should be helpful. That outer membrane is impermeable to water, so it has to have porins in it. What passageway is designed for water, as does the membrane in the inside, like a regular cell membrane. Whereas gram-negative cells have a cell membrane that water can pass through, and that a large amount of peptidoglycan is inherently porous. So know about peptidoglycan. NAMs and NAGs, thick in gram-positives, skinny in gram-negatives. Porous in gram-positives, so we don't worry about things getting through it. Held onto, just sitting on top of that outer membrane by those teichoic acids. And yes, everything else is about the same. So it's a good size target for identification and to attack. The gram-negative, a little harder to identify, a little harder to attack, but you can't broad brush and say one's worse than the other, because it all depends on what's going on inside. They all make exotoxins. That's how bacteria hurt you. The one that is inherently toxic is that lipid A of gram-negative. So staph and strep do not have endotoxins, where E. coli, gram-negative, other gram-negatives do, just because of their construction. That's there. So the NAMs and NAGs, the different thicknesses in gram-positive and gram-negatives, peptide interbridges and tetrapeptides for holding them together, the inherent porosity. Endospores. Again, it's not reproduction. Another question was about how they reproduce. Bacteria, for all intents and purposes, divide into binary fission. Yeah, there's some others. Eukarya, mostly mitosis or microbes, some meiosis, some have sexual reproduction. But they're different because of the nature of the DNA. Remember, circular DNA, supercoiled in bacteria, no nucleus. It's just held together in one close compact area. We call it a nucleoid. It looks like a nucleus, but it isn't one, because we don't have any membrane-bound organelles. 70S ribosomes. In eukarya, we have DNA in pairs, like we do. Have mitochondria. Plants have chloroplast and mitochondria. They're both about energy transformations. They're the heart of endosymbiotic theory. Endosymbiotic theory is just like a precursor cell swallowed a bacteria and said, you could help out here. I won't kill you if you just make energy. It's like, It's a beautiful thing. When I need more energy, you can reproduce. If I don't, you can't. Not that they talk to each other. Anthropomorphic bacteria, giving them. Endospores are an oddball because not many things do it. For our purposes, Clostridium and Bacillus. Those two genera, plural, genus, not geniuses. And remember, why is it not binary fission? Because when times are tough, not enough nutrients, carbon and nitrogen, not enough water, they split asymmetrically. The DNA is equal, but the cytoplasm isn't. The mother cell, the larger, engulfs the spore. The smaller cell, the DNA in the mother cell deteriorates. So it's not one makes two, one makes a different form of one. The two membranes coalesce together. In between them, peptidoglycan. Then they secrete dipicolinic acid. Calcium comes in, water is extruded. Protein layers called the cortex, maybe an exosporium, more protein, and that's why they're effectively indestructible. So that's other stuff that you need to remember. I had a question about flagella. Flagella are in eukaryotic cells, other than the protozoans, are relatively rare. We just have one in the male and the sperm cell. They're long intracellular appendages sheathed in membrane. That 9+2, they're made of microtubules, whether it's a little rotifer or something in pond water, or it's a sperm cell. Flagella and bacteria are common. That's how they get around, the ones that do. And they can, in the different arrangements, or they have peritrichus, monotrichus, lopho, amphitrichus, and basically they're attracted or repelled. It's very simple. Run, tumble, run. Yes.
One stick, like I know the flagella sticks out in one cell versus the other. Is it in prokaryotic cell that sticks out?
Yeah, and eukaryotic cells within the membrane. So it's sticking out, and it will spin in one direction to move it towards an attractant: chemical, air, heat, magnets, light, lots of things. Or the other way around. If it gets dark, it'll go the other direction. I'm not going there. Limited resources, that's where run, tumble, run comes in. I'll be talked out by the time I get home. Then my wife is going to make me do stuff. 70S for bacteria, 80S for us, but our mitochondria, endosymbiotic theory, have 70S. Eukaryotic microbes have membrane state, whether they have a cell wall or not. Plants and algae have sterols to stabilize the membrane. It helps. Fungi have ergosterol. We have cholesterol. So sterols are something that are unique to eukaryotic cells. We really don't see them in bacteria. Difference between fastidious and facultative. Fastidious require a lot of growth factors. Fastidious grow easy. Like E. coli, you give it, some water and a couple of salts and some sugar, it will grow forever. I mean, that's very facultative. It just grows like crazy. It's hard to stop it. That's why it contaminates a lot of things, staph and E. coli. So the thermal levels, you should know psychotrophs, the ones that grow in the fridge, 5 to maybe 30 degrees Celsius. Mesophiles, the big player for us, 25 to 45. Thermophiles, maybe like 25. I beg your pardon. Mesophiles are, again, 25 to 45, and thermophiles around overlap at 45 to about 70. Hot tubs, hot springs, that type of stuff. Those impact us. The extremes we don't reside in. We're not at the South Pole without something around us, because if you're not, it's pretty good. The fourth iteration of True Detective on HBO Max is all about endless nights got Jodie Foster. Weird. My wife and I are watching it. I can't say it's like everything. So, again, obligate aerobes have to have air. They have to have means to detoxify it with superoxide dismutase and catalase, the toxic derivatives of oxygen. Obligate anaerobes can't survive any oxygen, don't have those enzymes. Interestingly, the Clostridia, which are part of that, C. diff, tetanus, botulism, they are all endospore forms. and they're strict anaerobes. So it's dead, or it's dead tissue that they grow in because of the lack of oxygen. It's there. Refrigerator doesn't kill them. It's just slowing down spoilage organisms. Microaerophiles, just a little bit of oxygen. Flesh-eating bacteria and hot variant of strep mostly. And that H. pylori associated with gastric ulcers and gastric carcinoma. So those are significant, even though they're comparatively rare. And remember, we get rid of them by salting. Salt pulls the membrane away from the cell and it bursts, called plasmolysis. Almost done. Almost done. Almost done. Yes, one more thing. Growth curve. Lag phase, log phase, stationary phase, dead phase. Lag phase absorbs metabolites preparing to grow. Assume that if it's growing, it's not growing measurably. Anytime in a new environment, a couple of hours. Once it's ready to grow, it just grows. Everything is set, and they just grow continuously at a rapid rate, exponentially. That's that logarithmic graph, very, very sharp, steep slope. Got that all out. Two things. Best time to kill it, because it's voracious. It gobbles up anything in an environment. Toxins, antibiotics you throw in there, great. Also makes a particular waste product called a primary metabolite. That's where we get insulin and human growth hormone. It's a closed system, not the ones they use in pharmaceutical labs. But when we measured it, what happens is eventually the food runs out and there's so much waste, it impacts survival. At that point, because of diminishment of nutrients and available space, it enters another phase gradually, but it's a stationary phase as many as are living and dying. It's not zero growth, it's just equal growth and death. That's what makes it flat. Secondary metabolites, that's where bacteria that can make antibiotics because they're trying to survive. Survival of the fittest. They're a living thing. And at the same time, if they can make endospores, though it's limited, the ones that can do, they do so. Lastly, eventually the situation is just dire. More begin to die, and they fall off at the same steady logarithmic rate, although not as fast as they grow. That's it. You're done. Goodbye. See you Monday.
Are you gonna have this